Good morning, everybody. This is Jessie Maxfield from State Water Resources Control Board, and I'm here with Pete Ode, who's going to be presenting the technical refresher webinar for you all this morning. Pete probably doesn't need a lot of introduction, but he's really the lead on the technical team for this project, and for many of you, this presentation will probably seem familiar. For new staff, this is a great introduction to the technical work, and I'll let Pete take it from here. So we are recording this webinar, which is why we have you all on mute, so there will be plenty of time for question and answer when Pete's done with his presentation. So hold your questions, maybe write them down if you have any, and we will start Q&A probably at about 10:30 or so.
Hey everybody. I was looking back at my notes on this when I was putting some slides together and realized it's been 5 years since we started this project. In March of 2010, we had our very first kickoff meeting. So it's been quite an interesting ride. The technical work for this project was really wrapped up about 2.5 years ago, and at that point we started in earnest on the policy development. Since we've had Karen has left the project and we now have Jessie taking the lead, we thought it would be a good idea to give everybody a little refresher on technical work that serves as the foundation for the framework that Jessie's going to be developing. So, as Jessie said, it's going to be about 30 minutes of material here, maybe a little bit more, and then that should leave us plenty of time for discussion. So, hold your questions and we'll get to you when we get through. So, we start. If I get this thing to work. There we go.
Oh, yeah.
In case anyone is not muted, please mute your phones, and we'll the main presentation done. Thanks. The work I'm going to present here is really the work of a lot of different people. Many technical people are involved in this project. Ken at SWRCB and I were the co-technical leads for all of this. Andy at the Fish and Wildlife Bioassessment Lab and Rafi Mazur at SWRCB did a lot of the heavy lifting on the modeling. We had a scientific team that consisted of roughly 10 people that really contributed a lot at different times in this project. Throughout this effort, we were advised by a dream team of technical experts from around the country. We had some of the best stream ecologists and ecological modelers and bioassessment specialists from the nation advising this panel. So during the course of this project, we presented our plans to this group. They gave us great feedback. We then went back for more on the project, presented updates to them, and we did this about 4 times every 6 months or so. So it was a very intensive process. The group was just immensely valuable to us. We were really fortunate to have such a great group of people to work with. So I'll start here with the quick philosophy slide. The reason we're doing this work, or one of the key advantages of biological monitoring or biological measures of water quality, is that they provide something that we've never really had before, and that's direct measures of the key beneficial use, or aquatic life use, ecological uses. The traditional water quality measures get us by asking a question: can a water body support aquatic life uses? So we measure things like toxicity, pH, nutrients, pesticides, things like that. We'll ask whether if these meet our standards, or if they're out of compliance. Then we infer that maybe the biology might be impaired there. Trust, what we're doing with this project is creating a technical foundation for measuring the condition of aquatic life directly. So instead of asking, can they support the life use, the question is, does the water body actually support aquatic life uses? Our project started with a couple key objectives. The first is at the end of it, we want to have tools that would allow us to directly assess the weightable streams throughout the whole state. And to do it in a way that provided consistent meaning in different settings for both regulatory and non-regulatory applications. Our focus throughout this was on developing consistent tools, which is what I'll focus on too. But we also spent some time exploring causal assessment, which is the what happens next part. What do you do to identify impairment of the biology, then how to figure out what's causing it and what to do about it. So I'll come to that at the end. So California has a long-term vision for ecological indicators in which we would like to have multiple indicators of conditions, since we're really interested in ecological condition as a holistic concept. We're interested in measuring the health of invertebrates, algae, invertebrates, riparian vegetation, all of these things. I'd also like to be able to do this for the fleet of water body types that we have in the state. Large rivers, non-perennial streams, lakes, wetlands, etc. But for the project, we're starting with invertebrates and perennial streams. And one of the key reasons we're starting with invertebrates is that they're excellent indicators of general condition of these water bodies for a number of reasons. One, they're ubiquitous. You find these things everywhere. There's water. They're often very abundant and quite diverse. Because different species respond to different types of stresses, there's a lot of information in the organisms that we see at a site. So that's the basic concept. So about 10 years now, maybe closer to 15, the state has been started by developing standardized infrastructure for collecting, analyzing, and documenting this data. So we have standard methods in the state. This is the work of the Surface Water Ambient Monitoring Program, or SWAMP. So this is all this stuff is housed now. So we have standard feed methods, standard laboratory methods, standard data management and reporting, and also standard quality assurance. So that's the standards for collecting and analyzing the data. But to get to assessments, you need another step, and that's scoring condition from the data that you've collected. How do I turn then this list of animals that we see at a site into a score that tells us something about the health of that system, or that ecosystem? And there's really two parts to that, and that's what I'll focus on today. The first part is predicting expected biota. That is, in a natural state, what organisms should we expect to see at a site? If you think about California's diversity, it's a tremendously ecologically diverse state. It has continental scale diversity in our state. Think about all these different types of streams that we have. We don't expect that if humans had never existed here, we would still expect all these to have different sets of organisms that live here in each of these settings. And so any scoring tool we build has to accommodate that diversity. This is challenging, of course. There are strong natural gradients throughout the state, from geology, geologic variation, temperature variation, precipitation, elevation. There's a lot of different gradients that drive the biological diversity at these sites. So that's what we're focused on. The other challenge here is that California has regions of the state very high degrees of development, high urbanization, intensive agriculture, and that blight big sections of the state, and that makes things challenging as well. The basic approach here is called the reference site, or the reference condition approach. If we want to set expectations of what organisms should exist at different sites in a natural state, we want a network of streams that are more or less have low levels of human activity in them, but present all these different types of settings. So because we've been sampling so long, we had a pretty big data set to work with. We had about 2,400 sites to start with in our data set. And our goal was to screen this set and identify sites that had, that both represented the full range of stream types, but also where the biology was minimally influenced by stress, human sources of stress. The way we did this was we did a very extensive GIS exercise to screen sites. So for every site in that data set, that 2,400 sites, we calculated really everything we could get our hands on. Information about infrastructure in the watersheds, population, hydrification, land use, and additional information about fire history, the presence of dams and mines, and discharges and things like that. Put this all together and created a series of screens. We said you have greater than X amount of land use, then out. If you're higher than certain road density, then that site would be out. So we went through this times, and in the end, came with sites that passed our screens, and those are this list now marked in yellow. A pretty high number. We were pretty pleased with how many we were able to get, and it's with almost 600 sites that pass our screen. You'll notice maybe that pretty much all regions of the state that have water, or have perennial water anyway, are well represented except for the Central Valley, which has, I think, one reference site, and it is about feet from the next ecoregion, so it's as much in the Central Valley, but otherwise we have pretty good geographic coverage. Geographic coverage is just one of the things we care about. What we really make sure we weren't doing a lot of different sites of the same type. Relation sites, for example, that have low human activity in them, but don't necessarily represent the range of other types of streams in the state. So we looked at this a number of different ways, and here's an example. This cloud of dots are a set of 2,400 sites arrayed in origin. This way to think about this is that the axes on this are, let's see, stream size and precipitation on one, temperature and conductivity on another. There are a few other variables in there as well. But the simplest way to think about this is that sites that are close together on this chart are similar environmentally in terms of the natural attributes. Sites that are far away are not similar. And so if you think about the map on the left, the purple yellow dots are similar, are close together geographically, and it is far geographically. But when you plot them in this environmental space, red and purple dots are much more similar in the type of stream that they represent than yellow is the outlier in this case. That's why we do this. And what we're looking for when we put our reference sites against this is what we're hoping is that we have good coverage across the cloud, that we're representing the range of diversity. And the bigger gray circles here represent our reference sites. So we're pretty pleased with this overlap. If you look closely, you can find a few little gaps here and there, but for the most part, this is quite good coverage. So we were really happy with that. So the first part of the work was setting up that we represented the different types of streams in the state, reference sites. The second part is, did we sacrifice biological integrity to get it? Did we lower the bar, the criteria, or set the criteria such that we allow too much activity in these, what we're calling reference sites? And we tested that was by looking at a number of biological response metrics function of, for different categories. And so the three boxes in each of these plots are, left ones are the key ones. Those are, we took the reference sites, and we took one and divided them into two groups. Ones that had the least amount of human activity in them, and the ones that had the most. So they're all reference sites, but they're the sort of the dirty reference sites and the clean reference sites. They're all on the clean side, but dirtier. Clean and cleaner, I guess maybe it's a better way to say it. And contrasted those to the what had more stress in them. And so we're hoping, or what we would like to see, is that the reference groups, that there's no difference in biological scores, and that is clearly the case in everything we looked at. So we felt pretty good that the reference that we selected now both really good representation of the state, but also largely reference. So part one. Part two is turning this into a score. And so we used that work we just talked about as a foundation for an index that will allow us to compare the list of organisms we see at a site when we go to sample it, what we expected to see. So the tool we came up with or created is called California Stream Condition Index, or the CSCI. And it finds two different ways of converting a species list into a score. First, we call it a species loss component. And here we're looking at how complete is the taxonomic list, what we expected to see. The second one is ecological structure component, where we're looking at whether the ecological attributes of the organisms at the site are what we expect to see. Both account for natural sources of variation, and both measure different aspects of health. So we put them together for a stronger index. So a minute about the first one. So the species loss component is often referred to as observed to expected models or predicted models. Essentially what we're doing here is comparing the number of taxa we expect to see, we saw at a site, to the number of that expected, and that ratio tells us something about the condition. And we use reference sites now to tell us what is expected. So we compare sites to groups of similar reference sites, so what are similar to that test site, to determine what we should expect. So in this plot on the right, here are our reference sites now, and bubblegum colors here represent different groups of biologically similar groups, naturally similar groups. And so now the disparity then is defined by a series of natural gradients. So if you tell us the latitude, elevation, precipitation, temperature, and watershed area at your site, we'll tell you what organisms we expect to see there with a certain probability. Then we simply go ahead and compare. So most of the work in this is of course, in modeling E. We're predicting the probability of observing specific taxa at sites of similar environmental settings. An index score you can think of as an estimate of loss of native species. So if the index ranges from 0 to 1, you have a 0.8, you infer that 20% of the native species have been lost at that site. That's the true number. The second part is the ecological structure component. So here we do something, we use the same information, but now we convert the species list into a set of metrics that tell us something about the ecosystem function, diversity, sensitivity to stress. So metrics might be the number of mayfly taxa, the number of sediment-tolerant taxa, percent of herbivores, things like that. And because the expectation for these differs for different states, we adjust what the score should be based on where they are. So again, we model what the expected values for each of those metrics should be just like we did with the species list. Now we're doing it with metric. So plot of observed versus predicted values for this metric-sensitive insect species. So this is values expected at reference condition, because that's a one-to-one line in the middle there. As an example, consider two test sites. They have the same score, 30% sensitive species. In the first one, we predicted 30% and observed 30%, so that's right where we expect it to be. In the second case, we observed the same amount, but we expected 60%. So its metric score would be considerably lower. That's how just it works. The, of course, message here is that the score depends on the environmental setting and not just the metric value itself. So together, we put these two different measures together, and this is the simple average of the two is the CSCI. And that CSCI predicts, the simple way to think about it is it predicts the species and metric values to expect at a test site based on natural environmental factors. So you tell us a few things about your test site, and we'll tell which species and metrics we expect to see. So sample that and compare what you see to what you expect, and that is the CSCI. It ranges from 0 to more than 1. If OE is about 1, we infer that biological integrity is intact. If it's significantly less than 1, we infer that biological integrity is impaired or altered. So this plot is a frequency histogram distribution of the reference population. So scores at reference sites, they're not always 1. There's a range around that. The score CSCI ranges from 0 to a little more than 1. The mean is centered on 1. So we use property of that distribution then to help us set environment threshold. So when is biology impaired? The way we do this is we threshold, say the 10th percentile of the reference distribution. And that is likely intact, below that is likely altered. We can further out on the distribution, say if you're below the first percentile of that reference distribution, then you're very likely to have an altered community there. So exactly where these are thresholds that we used in our development. You know exactly where the thresholds are is something that is part of the policy development. So that's something that think about. But this gives you a framework for setting those criteria in an objective way. So last slides on this. The CSCI, we do a few things to confirm that it is a stable index. It does what we think it does. So we tested with independent data sets whether sites that we independently assessed as stressed or moderately stressed, as the CSCI scores do, and those distributions are the way we expect them to. We tested whether the CSCI varied was consistent across the state. We were really concerned about making sure that if we say, 0.8 in Southern California means the same thing in Northern California. And this is so we looked at reference sites, distribution of scores of reference sites throughout the state in different regions, and that's what this is a pretty good demonstration that those are pretty consistent. The last little test I'll show you here is a question of whether the CSCI changes over time. And throughout the data set, it's pretty stable. I should note that we haven't looked at the last few years. That's what we'll be looking at soon to confirm, but since we've been in extreme out here, it's a good time to look at that again. So stay tuned on that. We'll get back to you. To summarize, compared to other work that's gone on in the state, we have some big difference over what we had before. We set out to we had created indices in the past at different parts of the state, but they weren't consistent, right? And they were done differently for different purposes. So there's an effort to have one consistent set. So we started with a much bigger data set, much more rigorously screened, much more comprehensive assessment of integrity. We built in site-specific expectations much more than we did before, so that gave us a much better performance in our models. And you'll remember back from the beginning, a key was that we wanted to be able to assess all perennial wadeable streams in the state and for it to have consistent meaning. So we feel pretty good on how well we were able to do that. So that's most of the technical work. But remember in the beginning I said there was another aspect of this that we did spend some time on, and that's the area of causal assessment. We knew going into this that if we're going to be moving down the path toward measuring conditions with ecological tools, you figure out what we're going to do about it if you find impairment. So if you have chemical impairment, there's a pathway with TMDLs to figure out what the causes are, what the appropriate limits should be, and how to get there. It's a little different when biology is your endpoint, because the question is, there's some real challenges in doing that. So we needed to explore this, and this is not new. This is something that has a global attention. These tools are used widely throughout our country and around the world. So there's this field called causal assessment or stressor diagnosis, and a lot of that work has actually happened in our country, in our EPA, national EPA. We developed a framework that they call CADDIS, which is designed to do this. It's a causal analysis tool and this is one of the simple versions of their flow charts. Some of their stuff gets kind of crazy, but the kind of very established process that we wanted to explore. And so that tool, CADDIS, stands for Causal Analysis, Diagnosis, Decision Information System. Scientists at SWRCB have taken the lead on testing these methods in California. As part of this project, we put together some stakeholder scientists and regulatory teams to assess, just to pilot these, to test whether these methods would work in different parts of the state. And so we had some test cases in northern, central, and southern California. That work is documented in a guidance document that's on the Biointegrity website. The punchlines from that is that the tools look like they have some real promise for us in California, but we need some additional work and some tailoring to make them work here. So that is what the scientists have been working on the last couple of years now. And there's if you're interested, there's a NorCal CTAC short course that's being offered in about a month here at EPA. They'll put the link there so you can get to it, but they'll do a nice several-hour overview of all the stuff they've done, go into detail on some of these pilot studies that we did and some of the new work they're doing. Neat stuff. So I'll close with this. If you want more information on this, all of the documentation for the development of this process is pretty thoroughly documented on the Biological Integrity website. Every presentation that was given to the science panels, the report out from the science panels, meetings to the stakeholder and regulatory panels as well. All of those are there. So you can see in gory detail the development of how we got to that half-hour summary. So there's a lot there. There are two papers that describe the technical that are in both in press at Freshwater Science right now. They should be out soon. Probably by the end of the year we expect them. The scoring tool itself is although it's pretty complicated internally. We have some automated tools that we've been developing. Some early versions of those are available. Our goal is to develop full automation so that the scoring is easy for a lot of audience. And we just got funding available to scope that project, so we're starting down that path. I don't have a date yet for that, but check back in with us. We'll probably post progress on that on the website as well. And as I said before, for Causal assessment, it's also stuff on the website, but if you get a chance, check out the NorCal CTEC short course. And that is all I have for prepared material. Let me yell that out if people want it. And we can take questions.
Yeah, let me see. We need to, hang on, we need to figure out how to take your questions. So hold on one second.
Stop the sharing.
We can still keep sharing though. We can just go to, oh wait.
Okay. We need to get back to that main screen.
Hang on.
Let me stop sharing.
Okay. But I want to stop recording though. Is that going to stop recording? Okay.
So now you want to go back to participant.
Okay, we are unmuting.
It's still recording.
Yep, we're still recording. Okay.
So you can share again if you want to go back.
Okay.
To
Share my desktop. Okay, so can somebody say something so we know that you're not muted? Jen? It's like everybody's muted themselves now. Should I?
Okay, back to that participant.
Okay. Hang on. Unmute all? Okay. I'm unmuting you all. I have a question. Well, they only, oh, here we go. I hear somebody.
Good morning.
Good morning.
Yeah, good morning. This is Ed Strufenegger from the California Forestry Association. First, thank you for having this session. One question I have for Pete is in the development of the causal assessment. That was a major concern with a lot of the stakeholders, is that the CADDIS and the causal assessment process was very complicated, very expensive. Is there any work to be done on this to make this process a lot simpler for causal assessment?
Yeah, Ed, great question. For those of you who haven't been with us, Ed has been on our stakeholder panel since the beginning of this project, and you're absolutely right. One of the causal assessment work is part of this. One of the conclusions was that there's quite a bit of work to be done before it can be able to easily fit into programs. And this is the focus of the work that SWRCB is doing right now is to make that easier, to make more useful. So if you're able to make that the exact short course, that would be useful. I think I would contact. You could contact me if you want, and I can put you in touch with them, or you could contact Ken Schiff directly. They've been leading up the work to advance that.
Okay, thank you.
Looks like people are writing in some questions here, which is great. That makes it a little easier for us. One of the questions was, will we be posting the presentation on the Biological Integrity website? And the answer is yes. And we're also going to have Eric Burrs put this recording of the webinar on the YouTube channel. I'll make sure to pass that to everybody so that you can share that. Yeah, so is it the Water Quality YouTube channel? Or? Okay. So we'll share that information, but we'll also post the presentation as well. Okay, can you see the questions there?
I can see some of them.
Okay, so we have one about
Okay.
yeah.
So, a question from Ross. Question is, would this be appropriate for assessment of marine life for intake, say, desalination or discharge in an ocean and bay environment? Well, the technique certainly would. The work that has been worked on by our group is freshwater focused, but the group, SWRCB, who's working on it, has a strong marine component. So I would encourage you to contact them and see if they've thought about it. My guess is that they have, but I can't speak to that directly.
Yeah, they can't, those are private messages, so. A question from P. Church. Why were the IBIs discontinued? And for those of us who may have used IBIs in the past, can you discuss how those IBIs relate to the new CSCI?
So, IBIs you can score your, because they collect the data, you can score them with any pretty much tool that you've used in the past if you want to. We don't recommend using them anymore because what we've created is a much more precise, much more accurate tool and have the benefit of being consistent. So the initial tools I would think of as early development. These should replace those. But really are dying to use the old ones if, say, you want to compare your old scores to scores. That is certainly possible.
So the next question is from Whitney Brennan. Is there a list of reference streams used available? How do we do CSCI if the stream we are assessing is already used as a reference stream?
Okay, first of all, there is the list of all these reference sites is posted on the Bioassessment website. That's all there. And the fact that a site might have been used as a reference stream has nothing to impair your ability to use the CSCI at that site. You can still use it. So that we have on the chat comments.
Did anybody want to ask questions out there?
Jessie, this is Rachel Kubiak. Can you hear me?
Yes, we can.
Okay, I have a quick question about a couple of, this is Rachel Kubiak with Western Plant Health Association. And I participated in the meeting, I think it was in January, and there was a couple of things that were discussed which are more policy, I guess, questions or issues than they are technical, but two had to do with sort of the scope. And that originally, number one is that originally there was a focus on the perennial weightable streams, and that has now changed to sampleable streams. I'm curious if, well, the other being was that initially when we met one stakeholder groups last year, that the direction was that there wouldn't be a requirement for use of the CSCI index. That changed also at the January meeting. So my question is, will be a forum to have further discussions on those items, or is there any idea on and that will be discussed? Because I know, for example, in the agricultural world, there's concern about that now. In the environment where constructed conveyances and other things would not be discussion, and there's concern on that. Since there's been, or it looks like in the focus or the scope, there's been changes, will those issues be addressed in one of the stakeholder groups in the near future, or through other means, or is there any, do you have any idea on that?
Let me start because there's a little bit of technical in there, and then I'll hand it over to Jessie for the more policy aspects of your question. So the question about perennial versus sampleable. So the scope hasn't really changed. It's acknowledgement. The wording more reflects an acknowledgement of the fact that the depth of perennial is not a very useful construct when trying to figure out what streams these tools apply to. Because low permanence is not a dichotomy between perennial and non-perennial. It's actually a continuum of how permanent. As you can see in drought conditions like this, streams that might have water flowing in them year-round for decades may have dry periods. So what we've done some work on this that these tools work really well in streams that have water in them most of the year. And so the definition we're going with is more practical one than a theoretical one, and that seems to work better. I'll let Jessie handle the requirement. That was the original plan was to define the type of a tool that would be acceptable. I think there was a legal problem with using that, which is why they want to specify this tool itself. But Jessie may know more about that than I do.
Yeah, I don't really have anything to add to that. I think we want to pick the tool that is most appropriate to use, and this is currently the tool that we feel is best, and it's a consistent statewide approach. But in terms of how that's going to be written into the plan, I'm not sure about that at this point.
But you have more stakeholders.
Yeah, and that was the other thing I was going to add, is that right now I'm in the process of still trying to familiarize myself with kind of where we're at in terms of how things left off with the different stakeholder groups. And my plan is to gather some focus groups over the summer to try to answer some of the remaining issues that we have and address the remaining issues that are kind of still on the table in terms of making some decisions about what direction we want to go in. And I'm trying to prepare some to bring to the focus group so that we can make the most use of everybody's time. And I will be talking to some people who have let me know that they're interested in being part of those stakeholder groups in order to help me to decide who to invite to those meetings, and also just sort of what we still need to figure out and what the best way to go about doing that will be. But it's looking like we're going to start holding some of those meetings in June, so I will be getting in touch with folks about that and putting out messages on the LIRIS. Does that answer your question? Or?
Yeah, I think it does. I think they're still, my understanding of this, and the reason I tuned in is because I've only been involved in this for probably the last year. But the sense I've gotten from other folks in the ag world is that including ag drains or potentially including ag drains or other type conveyances was not that wasn't communicated as part of the intent. And at a meeting in January, it was specific for us that would be included somehow, and that came across as being established. And my understanding is that historically that wasn't necessarily true. So, as long as there's going to be meetings in June, I think.
That's probably going into more details about the implementation plan itself, and I don't think any decisions have been made at that level at this time.
Okay.
I just want to, we can talk more about this, Rachel, if you want to talk to me, but I want to focus the remainder of our time on asking any questions about Pete's technical presentation. And we do a few more that came in here on the chat. Yeah, sorry. Okay, let's see. We had a follow-up from Michael Ross. Where is the TDS cutoff for this freshwater-centric assessment? What about the delta or other brackish systems?
Yeah, so this is intended for freshwater systems. We did not define a cutoff for that though. So that would be willing to open up and think about doing though. I don't see a problem with doing that. But just to be clear, the tools are not intended to apply brackish systems.
And there was a follow-up from T. Church about the IBIs. Do the IBIs correlate with the CSCI conditions?
Yeah, very closely.
I don't mind that I'm reading your questions as they're written and saying who you are. Because nobody can see these, right? Is that at least a minute? Okay. Okay, let's see. Dan, ignorance, new to this. Is this new biointegrity measure intended to replace or be in addition to the standard pollutant TDL measures of impairment?
In addition to.
Good question. Okay, let's see. And does the CSCI consider best attainable condition? For example, our urban reference reaches?
Okay, good, great question. And the policy part has spent a lot of time talking about this. But from a technical perspective, the CSCI itself does not incorporate best attainable as part of its development. You use the CSCI anywhere, and you can set thresholds for different types of settings, and that is certainly a valid subject for discussion in the implementation development, and that was a large portion of the discussions that have happened so far. So if you go back to all these presentations that have been on the websites for this from the policy development cover this quite a bit. So but again, for the technical part itself, the CSCI, the referees are strictly low human activity sites, because we want to assure that that the expectation is based on what should be there naturally.
Okay. You see the one from Mike. There was another comment from Michael about ag grains having T at 8,000. Maybe as high as 8,000. I think it must be related with
yeah.
It appears that the applicability of this tool requires defining perennial streams. Will regional boards need to define perennial streams in the basin plans?
I'll say, and this is probably a question for you, but not for this discussion. I mean, essentially that is you're right that there'll have to be a definition of applicability in the implementation plan.
That's one of the issues that remains to be addressed, and one of the things that we probably would be talking about in the focus group. Things like that, those are the types of issues that are still on the table. RCI and CADDIS applicable to effluent-dominated streams. Got a message, Michael, about being on the list for interested parties. Okay, thank you. Let's see. Can you expand on the project to develop automation tools for CSCI? What is the likelihood of funding for this?
Actually, we almost got it funded entirely, but there were some new regulations in state government that influence IT-related project funding. So work was funding for the scoping, and we hope to get funding after that scoping phase is done, when we think the probability of getting funding for the automation tool.
My opinion is the likelihood is good, based on what I've heard. So. Getting the messages about being added to different focus group lists, and thank you. I will get that all. I have to make sure I know. If you want to email me directly about that, this is Jessie Maxfield, because I don't know that I can figure out who you are based on your name here. Okay. I get your email, but if you don't mind emailing me directly, that's probably the way. They'll be able to have a record of this, right? Of you chat questions? Okay. The WebEx guy, so. Sorry.
Give them your address, right?
Yeah, oh, my email address?
Yeah.
My email address is jmaxfield, JMAXFIELD @waterboards.ca.gov. One who sends out everything on the LIRIS, and my email is in the message that comes out. So at the bottom of the Lyris and the signature, you can find all my contact information. Were there any questions on the phone? You have to unmute yourself. I'm not hearing any. I think we got all the ones on the chat. Well, thank you all for joining us today. You can follow up with any emails if you have questions. But thank you for joining us, and we will let you know where to find the presentation and the webinar recording when it's available. Everyone.
Thank you.